Hello everyone, my name is Jiayu Shang. Today I will give you a presentation about our recent research, bacteriophage classification using Graph Convolutional Network. First, let me introduce what is bacteriophage. Bacteriophages, or phages for short, are the most common and diverse entity in the biosphere. They are the most dangerous killers in the world and kill trillions a day. But fortunately, rather than infect humans, they mainly infect bacteria and archaea. Here I show you a figure about the E. coli bacteria that was infected by the T2 phages. But you might ask, why we need to study phage? During the life cycle of the phages will infect the bacteria and replicate within them. Then they will kill the bacteria and release their offspring. So scientists thought phages can be used as an antibiotic to treat the disease caused by the bacteria. Also, you can see antibiotics may kill different bacteria in such a region, but phages will target the bacteria that they mainly infect. What's more, there are some bacteria that are resistant to the antibiotics, which we call superbugs. So if we can find the phages that infect the superbugs, the phages might help us to kill them. To use the phages as our super weapon, the first thing we need to do is to group the phages with the same biological function. And that's so-called phage classification problem. In the phage classification task, the initial dataset are DNA sequence. What we want to do is to group different types of phages together. Here I show you 4 different kinds of phages on the virus. They have different biological functions, which means they might infect different bacteria. However, there are many challenges for the phage classification. The first thing is, because we know phages are diverse, there are many new phages with new proteins. For example, 60% of the proteins are hypothetical proteins, which means we don't even know their functions. Over 10% proteins don't have the alignment results, which means they are totally new to the database. Also, phages from different taxonomic groups share similar proteins, and this will be a hard case for the classification tools to separate them. The second challenge is the number of the known phages is just the tip of the iceberg. As you can see in Figure 1, the number of the newly identified phages increases largely every year. But if we use the traditional alignment-based method, the limited known phages will cause this method hardly identify the new phages. That's why we need to design a new method. In our research, we use the semi-supervised learning method, utilizing both labeled and unlabeled samples to enhance the learning ability. We construct a knowledge graph and apply the Graph Convolutional Network. As you can see in the knowledge graph, the nodes represent the phages either with label or without label. The edges between nodes represent the similarity between the phage pairs. Then, Graph Convolutional Network can measure the relationship between different nodes and predict for the unlabeled nodes. Next, I will show you how we construct this knowledge graph. First is the age. The definition of the age is the similarity. There are 2 types of similarity we consider. The first one is the similarity of the protein organization in the input phages. Intuitively, if 2 sequences share similar protein organization, this sequence pair will have a higher probability belong to the same group. As you can see in this pipeline, we use the Markov chain algorithm to cluster proteins. Each protein cluster contains similar proteins. Then we can build a lookup table for each phage. For example, for phage A, it occupies 4 different protein clusters, and for phage B, it also occupies 4 different protein clusters. Phage A and phage B share 3 different protein clusters, and the total number of the protein cluster is 8. Then we can use the equation to calculate the probability that 2 sequences share at least C common protein clusters. This equation calculates the background probability, and thus, the smaller the number, the more significant the similarity. Because in this type of similarity, we only care about the number of the protein clusters. So in the next step, we will also measure whether 2 phages share high overall sequence similarity. If 2 sequences share high similarity, they also have higher probability belong to the same group. To sum up, we consider both protein organizations and the sequence similarity in the age construction. But what about the nodes? The nodes feature encoding is used a pre-trained CNN model. And as you can see in figure 3, the pre-trained model will capture motif-related patterns from DNA sequence. This is a multiple filter size convolutional layer because we want to capture different lengths of the motifs. Also, since the input contigs might have different lengths, we cut each contigs into 2 kbp segments. Then, we will calculate the average encoded features of segments from the same contigs. Finally, we have the knowledge graph. As you can see, the green nodes, red nodes, and the blue nodes represent the labeled nodes, which means those sequences have known taxonomic labels. And the nodes in gray represent unlabeled nodes. During the graph convolutional neural network, in the training mode, we will minimize the loss for the labeled nodes and update the parameter in the backpropagation. And in the testing mode, we will predict the unlabeled nodes. But you might ask, how we can use these unlabeled nodes when training? Let me show you the key insights of the graph convolutional network. For example, let's call this unlabeled node the current node. In the first convolutional layer, we will use the information delivered from its one-step neighborhood to encode a new node feature. And we will use this new node feature as input for the next layer. Also, the convolutional layer will calculate the information for each node in the knowledge graph. Thus, in the second layer, the two-step neighborhood information will also be delivered to the current node. Because we minimize the loss for the labeled nodes, the label information will be backpropagated to the unlabeled nodes, and thus we can make a prediction. Next, I will show you some exciting results in our experiments. In the experiment design, we use 4 different metrics to evaluate our performance. Because the dataset in the bioinformatics field are usually imbalanced, we use the macro matrix to treat each class equally. We also define the prediction rate, which means how many of the inputs are predicted. Then, we test our method phage-GCN on different datasets: the simulated data, the real sequencing data, and one metagenomic data. We also compare our method with 3 state-of-the-art tools. 2 of them are learning-based methods, which means they use the machine learning algorithms. And the POGs is using the alignment-based method. In the simulated data, all the testing phages are removed from the dataset, so we can treat them as new phages. Then, we randomly simulate contigs in 3 different length ranges. The results shown in figure 5 reveals that with the increase of the length range, the performance becomes better. This is because we have more information from the longer contigs. The light blue line shows the result of the phage GCN, which is much better than other tools. Also in figure 6, the total bar shows the prediction rate, and the bar with texture shows the accuracy. In this figure, phage GCN also achieved the best results across different length ranges. In the real sequencing data, one of the data we used was downloaded from the NCBI. Since it's a reads dataset, we assembled the reads into contigs first. And because we want to evaluate the performance, we need to know the exact real labels. We run the BLASTn to reassign the labels for the contigs, and the table 1 shows all the reference genomes. We remove all these reference genomes in the model database to treat them as new phages, and then we predict all these contigs. As you can see in figure 7, the performance shows that phage GCN can not only find all the contigs, but also achieves the best performance. All the phages are predicted correctly. In the final dataset, we use the oyster metagenomic data to test our model. But because this is a new dataset, we don't have the reference genomes for the contigs, which means we don't have the ground truth. We compare the result with the vContact because it has the highest precision compared to other methods. The result shown in table 3 reveals that phage GCN can largely improve the composition analysis, and also all the contigs predicted by the vContact are predicted by the phage GCN with the same labels. Notice that phage GCN achieved higher precision than vContact in the previous experiments. And thus, we thought the prediction of the phage GCN is reliable. To draw a conclusion, first, we construct a knowledge graph and we apply GCN to utilize both labeled and unlabeled data for training. In the construction of the knowledge graph, we combine the strengths of the reference-based method and the learning-based method. Finally, in the future work, we are going to extend phage GCN to detect phages for more complicated metagenomic data. And that's all for this presentation. Thanks for listening.